Hello, my name is Eve Marder, and I'm head of the Division of Science at Brandeis University and professor in the biology department. And this is segment 2 of my 4-segment series, and I'd like to focus today, or in this segment, on principles of neuromodulation. I'm going to show you examples from the crustacean stomatogastric ganglion and nervous system, but I think everything I'm showing you today is general to all nervous systems, whatever they are. So to start off, I'd just like to show you an overview of the stomatogastric nervous system. The stomatogastric ganglion is seen here in the center. It contains 30 neurons, but the important thing is it receives descending modulatory inputs from 25 pairs of neurons with somata in the paired commisural ganglia, or COGs, and the single esophageal ganglion. Those inputs travel down the stomatogastric nerve and ramify in the neuropil of the stomatogastric ganglion. When these ganglia are left attached to the stomatogastric nervous system, you see the two major motor patterns of the stomatogastric nervous system: the slow gastric mill rhythm that you can see here with alternations between LG and interneuron 1, and the faster pyloric rhythm seen down here in the intracellular recording from the PD. Now, for many, many years, my laboratory and many other laboratories around the world have been trying to describe the full complement of neuromodulatory substances and their actions in the stomatogastric nervous system. My interest in this goes back to my days as a graduate student, when people all over the world were studying different neurotransmitters and different neuromodulators. So person X was studying serotonin, and person Y was studying GABA, person Z was studying glycine, and I felt it would not be possible to understand why there were so many neurotransmitters and neuromodulators in the nervous system until we had the full cast of characters, or the full set of players, and could understand the organization of the modulatory control in a given nervous system. So I set out very early on to try and get all of the molecules that would be possibly used as neurotransmitters and modulators within a simple, small nervous system to try and see what kinds of general principles would arise. So in the early days, we mostly started out doing immunocytochemistry. A peptide or an amine would be isolated, someone would make an antibody against it, and then we would use whole mount immunocytochemistry to study its distribution, presence and distribution in the stomatogastric nervous system. And this is what those data look like. This just happens to be an antibody that was raised against B-type allatostatins. The identity of the peptide is not terribly important, but you can see that there are beautiful cells that we're staining in those anterior ganglia, and then projections into the stomatogastric ganglia that I'll show you here. And on the left, you see that stained neuropil projections in the stomatogastric ganglion itself. And over here, there's a diagram that shows the distribution of the allatostatin B projections that from the commisural ganglion, the esophageal ganglion, and arising in the stomatogastric nervous system. Now, this was done with immunocytochemistry, but there's always a problem in using an antibody, which is you actually never know that the peptide you're seeing is identical to the peptide the antibody was raised against. And so for many years, we struggled with this, and we tried to purify peptides. And then the revolution in MALDI Mass Spectrometry came along, and it greatly simplified our lives. A number of people working in crustaceans, including Lin Jun Lee and Beth Stimler, started identifying the peptides in the stomatogastric nervous system by MALDI, and these just happen to be spectra. And on the basis of this, it turns out that there are a number of different peptides that are found as members of very large peptide families. So, for example, there are 8 or 10 different allatostatin B peptides in the stomatogastric nervous system. And this becomes very important because if you want to do physiology, you actually need to know the real structure of the peptide. So the next slide just shows you what we now know about the modulatory control in the stomatogastric nervous system. So over here you see the neuropil of the stomatogastric ganglion, And the stomatogastric nerve, you see all of these colored lines. These are descending modulatory inputs, and found in those are substances like acetylcholine, GABA, nitrous oxide releasing neurons, dopamine, histamine, octopamine, and a raft of neuropeptides. And you can see them here. That's the ASTB I just showed you before, and a ton of other peptides. And some of these are found in single copies. For example, Proctolin is a peptide that's only found in one form. Many others have very large family members. Also important is a very large number of neuropeptides that are secreted from the pericardial organ, which is a neurosecretory structure, into the hemolymph, and then the blood carries that to the stomatogastric ganglion. So neuropeptides and other modulators reach the stomatogastric ganglion from the hemolymph and from these descending modulatory inputs. So the important lesson here is that there's not one modulator, there are not two modulators, there are not three, they're a very large number. And I believe that this lesson will be true of any circuit that we study in any animal once people get down and dirty and actually do the work to study the distribution of all these substances in any region of the brain. So we have this richness of modulator potential action. Now, what do neuromodulators do? Most of you probably know that a neuromodulator is often defined as a substance that binds to a receptor, triggers some sort of second messenger activity, and eventually alters some membrane conductance, either to modulate synaptic processing or one of the voltage-dependent currents in a cell. Here's just an example of how profound the effect of a modulator can be. On the top trace, you can see in normal saline, an action potential in the LP produces virtually nothing in this case, in this preparation, in the PD cell. But after Václav Thermolai applied the peptide, red pigment concentrating hormone, or RPCH, you can see each action potential LP produces a very large iPSP in the PD cell. So this is an example of a synapse which is there anatomically, but functionally virtually invisible until the modulatory environment has been altered, so that the modulator then, if you will, enables or creates the functional synapse from the anatomical potential that was there. Modulators can also very strongly alter the intrinsic membrane properties of cells. Now, these are old recordings that Judith Eisen made many, many years ago. And what she did is she isolated an AB cell, and then she washed all the modulatory inputs away. And you can see at the beginning of the trace, the cell was silent. And then here at the downward triangle, she applied pilocarpine, which is a muscarinic agonist. And slowly over time, you can see the beginnings of oscillations. And then they got faster and larger. And then she pushed the button on the chart recorder to make the speed of the chart recorder increase. And now you can see these beautiful depolarizations and hyperpolarizations of the cell. And then she washed it away, and she did the same thing with serotonin, or 5-HT. And again, you can see activation of the burst in a previously silent cell. So this again shows you how profound the actions of modulators can be. They can qualitatively change the properties of a cell, in this case from being silent to being very, very strongly oscillatory. Now, a few more sort of pieces of information about neuromodulators. These are recordings that Andy Swenson made on the LP cell in the crab stomatogastric ganglion, and he filled a puffer pipette with all these different substances, and he applied these substances to the cell. And you can see that each one of them depolarized the cell, made it fire action potentials, and to some degree made it more bursty. And they had very similar actions. Previously, Jorge Goawash had characterized the properties of one of those peptides. I had mentioned this before, Proctolin. And now these are recordings made in voltage clamps, so you're looking at inward currents. And the important thing here is that this at minus 40 millivolts, you can see a large inward current, but at minus 60 millivolts, the proctolin produces a smaller current. So this peptide activates a voltage-dependent inward current, which tends to make the cells more excitable and actually has a current voltage curve almost identical to that of the very famous NMDA receptor that you find in vertebrates. And this voltage dependence turns out to explain the way this peptide activates neurons. But the reason I'm bothering to show you this is to show you another feature of this, which is that many of the modulators, peptide modulators in particular, converge on the same final current. Each one of them activates a different receptor, but they can occlude each other's activity because they converge on the same voltage-dependent current. And so this is shown in the experiment shown here, where on top up in A, now we're in voltage clamp, so you're looking at an inward current, which is evoked by proctolin. And then what Andy did is he put this second peptide, CABTERP, in the bath, and you can see a large inward current. And then he reapplied proctolin, and you can see that there's very little effect, additional effect. And then he did the experiment in the opposite order. He puffed on CABTERP, got an inward current. Then he put proctolin in the bath, and he occluded the effects of the CABTERP. So this is telling you that they're occluding each other's action at the level of downstream of the receptor because they're completely structurally different peptides. They bind to different receptors, but they appear to be converging at the level of their final target. And this may be very, very, very important for us to eventually understand how circuits can be stable despite all the modulation you see. Now, just some summaries of work that comes from work from our lab, from Mike Nussbaum's lab, from Ron Harris-Warwick's lab. In particular, this work on dopamine came from Ron Harris-Warwick's lab. What Ron did is he characterized the dopamine responses on each of the neurons in the stomatogastric ganglion, and he asked which currents were the direct targets of dopamine. And every cell had multiple targets for dopamine action, but they were different. So you can see that this is a very complex sort of set of membrane targets for the actions of a single modulator. And then this slide shows, and this came from work in our lab mostly, that a single neuron, here the LP neuron, has receptors to a very large number of different modulators. So that tells you how sort of pleiotropic these effects can be. Now, the take-home lesson from what we know, this now is the larger pyloric circuit. Each neuron has multiple currents subject to modulation by many substances. Each synapse is subject to modulation by one or more substances. So now this creates a problem. Anybody who has ever played with any models of complex systems knows as you start playing with the parameters in a model, the first thing the model does is it crashes. However, when we study these modulators, they often don't crash, or the networks don't crash very much. When we first got these data, we sort of said, oh, well, since every modulator is acting on a different subset of potential targets, we would expect that every modulator might produce a different form or a different kind of pyloric rhythm. And that's what you see here. This now goes back to Jim Wyman's days in the lab many years ago, where what Jim did is he removed all the modulatory inputs, and then he washed the preparation extensively, and you see he lost all rhythmic activity, and then he applied here pilocarpine, and he got back a beautiful pyloric rhythm. He washed, and then he applied serotonin, and he got back a slightly different kind of pyloric rhythm, and then he washed, and then he applied Proctolin, and he got a pyloric rhythm. He then washed and then applied dopamine, and now you can see what I would call the only of these modulators that produces what I would call a crash, where you've lost the functional alternation between LP and PD. And this only happens at high concentrations. And then Jim washed again. He applied a few more peptides. And in each case, you can see a different form of the pyloric rhythm. And so when we first got these data, we sort of said, oh, this really makes sense, because now we can understand how the same circuit can be tuned to produce different variants of basically a motor pattern. And that still holds. Today I look at this and I see something a little bit different. Today I look at this and I wonder why it is that so many of these peptides and other modulators are producing perfectly stable motor patterns, because despite the fact that there's been so much modulation of so many parameters in the circuit. So looking at these recordings, you realize, or you will have realized, that these were all made by using exogenous applications of neurotransmitter in the bath to an in vitro preparation. But that's not the way neuromodulators are usually applied by the animal. In the animal, neuromodulators are released by modulatory neurons that contain those modulators, often in the presence of a co-transmitter. So Mike Nussbaum's lab has spent many years trying to find and characterize the descending modulatory neurons and then figure out what their co-transmitter complements are. He's then studied these one by one, and I'm going to show you some of those results. Here, this slide describes the effects of stimulating two different modulatory neurons that contain the peptide proctolin. So on the left, you see recordings from Nussbaum's lab showing that from the DGN, the dorsal gastric, the gastric rhythm is off. LG is also silent, but the pyloric rhythm is on. That's in control. And then they stimulated MPN, that's modulatory proctolin-containing neuron. And that's seen there on the bottom trace. And now you can see a very strong activation of the pyloric rhythm, but no activation of the gastric mill rhythm. On the last panel, they stimulated a neuron called MCN1. And here you can see that that proctolin-containing neuron now activated the gastric mill rhythm, as well as made some modifications in the pyloric rhythm. And the next slide shows a third proctolin-containing neuron, MCN7, and its actions. Here, strong depolarization of MCN1 activated some gastric activity and strongly influenced the action of IC. It turned it from being purely pyloric, as you see here, to now firing in gastric time, just as I showed you in my first talk about other neurons that switch from pyloric to gastric behavior. And this is all summarized in this diagram, which shows you that the three different proctolin-containing neurons produce different modifications of the STG motor pattern: MCN1, MPN, and MCN7. Now, we know that MCN1 contains proctolin plus GABA plus CAB-TERP. MPN contains GABA and proctolin. MCN7 does not contain either GABA or CAB-TERP, but does contain proctolin and may also contain something else. But be that as it may, proctolin can be delivered to the STG neuropill via these three different sets of neurons and these neurons can still produce very, very different motor patterns because of the differences in their co-transmitter complement, as well as the local circuitry that's found there. Now, to summarize what we know about neuromodulation, I'd like to say that the connectivity diagram, or the connectome, presents a series of possibilities, possible things that the circuit can do. But circuits are now reconfigured by the temporal dynamics, that is to say, how they're working, and the modulatory environment. And it's the temporal dynamics in the modulatory environment that actually construct the set of parameters that make the functional circuit that is operating as the circuit is running. They do that by modulating the neuronal excitability and by modifying synaptic strength. So it's those synaptic strengths and neuronal excitability levels that are occurring in that modulatory environment that specify the circuit that is producing that behavior. Now, the conundrum for the future is really how can circuits be reliably modulated without running the risk of overmodulation? And I think if we think towards trying to understand problems like addiction and mental illness, we come right up against the threshold of trying to understand when neuromodulatory processes keep circuits in the place where they can be modified and modulated in a constructive way to where you cross the boundary between appropriate modulation and overmodulation, or inappropriate modulation, and then you get loss of circuit function. And I would just like to again acknowledge the wonderful people in my lab who have over the years participated in all this work. Thanks.